For this video, we will cover how to complete one-trait Punnett squares, specifically those pertaining to traits with incomplete dominance. Now, to explain the difference between true dominance and incomplete dominance. True dominance is where we have either a dominant or recessive version of the trait. Short versus long fingers could be a good example. In this case, if we use A's to show the genotypes, short fingers are dominant to long fingers. So a homozygous dominant individual, or big A, would have short fingers. A heterozygous individual, or big A, would have short fingers. And a homozygous recessive individual, or little a, would have long fingers. Now, with incomplete dominance, we can actually have a third or intermediate phenotype. So here we could use straight hair versus curly hair. And note that in this case, there isn't a trait that is dominant to each other, but we can still use upper and lowercase letters to indicate the specific traits. So let's say that big A is straight hair and little a is curly hair. Here, an individual that is homozygous dominant would be big A, and have straight hair. An individual that is homozygous recessive would be little a, and have curly hair. Now, if we have a heterozygous individual that is big A, then it would be an intermediate between the two, and this individual would have wavy hair. Another way that we can display this is by using ones and twos. Here we could have a big A1 and a big A2. Big A1 means straight hair, and big A2 means curly hair. So an individual with two big A1s would have straight hair, and an individual with two big A2s would have curly hair. And an individual with a big A1 and a big A2 would have wavy hair. I tend to prefer the first option, so that is what we will use in this video. Alright, that was a long explanation on how the incomplete dominants work, so let's try and solve some Punnett squares. Let's start with two heterozygous individuals that have wavy hair. These would both be big A, little a. Let's make our square, let's add in the potential alleles, and let's multiply the alleles into the square, and we should get top left is big A. Top right is big A, little a. Bottom left is big A, little a. And bottom right is little a. At this point, we can now start asking our questions about genotypes and phenotypes. So let's start with, what is the genotypic ratio of my cross? Here I'm just asking about the number of homozygous dominant to heterozygous to homozygous recessive individuals. Now if I look at my cross, I can see that I have one dominant individual, two heterozygous individuals, and one recessive individual. So my ratio comes out to be 1 to 2 to 1. Next I can ask, what is the percent chance that a specific genotype is seen? Since I have 4 boxes and those equal 100%, each box equals 25%. So if I have one box with a dominant individual, that will be 25%. My 2 boxes for the heterozygous will be 25 plus 25%, equaling 50%. And my 1 box for the recessive individual will be the last 25%. Keep in mind that the math has to add up to 100%. So if it does not, go back and relook at the math to see where the error is. Next, I can ask about my phenotypic ratio. And here I'm simply asking about what is the number of straight hairs to wavy hairs to curly hair individuals. So if we look at the square, we have one dominant individual, which means one straight hair individual. We have 2 heterozygous individuals, which means 2 wavy hair individuals. And we have one recessive individual, which means one curly hair individual. So our phenotypic ratio is also 1 to 2 to 1. We can then ask about the percentages of each phenotype. And since both the phenotypic and genotypic ratios are the same, we get the same percentages again. So our dominant or straight hair individuals will be 25%. Our heterozygous or wavy hair will be 50%. Our recessive or curly hair individuals will be 25%. Again, we can make sure it adds up to 100% to check our math. Let's go ahead and reset our square so that we can do another example. We're still going to use hair types, but we will use different parents for our cross. Let's go ahead and use a straight-haired male, big A, with a wavy female, big A, little a. We set up the square, let's add in the potential alleles, let's multiply them, and then we get top left as big A, top right as big A, little a, bottom left as big A, and bottom right as big A, little a. Let's do the genotype math. We have 2 dominant individuals, 2 heterozygous individuals, and 0 recessive individuals. This gives us a ratio of 2 to 0. Note that while the 0 can be assumed, we generally include it for good practice. In terms of the percentages, it comes out to be 50% dominant, 50% heterozygous, and 0% recessive. Now let's do the phenotypic math. Here we have 2 individuals with straight hair, 2 individuals with wavy hair, and 0 individuals with curly hair. So the ratio is also 2 to 0. Our percentages are also going to look the same. 50% dominant or straight hair, 50% heterozygous or wavy hair, and 0% recessive or curly hair. As our examples here have shown, it is very common with incomplete dominance to get the same genotypic and phenotypic ratios and percentages. So just keep that in mind as we're completing our genetic problems. As you go through and complete these types of Punnett squares, make sure that you follow the basic rules for setting them up. Set up the square and add in the potential alleles from each member. Multiply the alleles against each other, then do the math for both the genotypic and phenotypic parts of the problem. This concludes our video covering how to complete one-trait Punnett squares, specifically those pertaining to traits with incomplete dominance.